Okay, so 1620 is our one extra spicy problem for this week, and okay, I've been doing these problems for I don't know, about 3.5 hours now, so it looks like a long one too, but hopefully we can get through this relatively quickly. Let's take a look. Start by writing the number here, 16.20. In a theoretical operon, genes A, B, C, and D represent the repressor gene, the promoter sequence, the operator gene, and the structural gene, but not necessarily in the order named. Okay, so we are working with a theoretical operon. Okay, and it expresses 4 genes, A, B, C, and D, and I guess the repressor gene doesn't have to be part of the operon. But okay, what are the components? We'll say components are repressor, promoter, operator, and 1 structural gene. Okay, so 4 genes, 4 different things. A could be a repressor, it could be the promoter, operator, or structural gene. So could B could be one of these, C could be one of these, D could be one of these. We don't know what they are yet. I guess we're going to have to figure out which goes to which in this problem. So this operon is concerned with the metabolism of a theoretical molecule called TM for theoretical molecule. From the data provided in the accompanying table, first decide whether the operon is inducible or repressible. Okay, is the operon inducible, like the lac operon, or repressible, like the trip operon? Then assign A, B, C, and D to the 4 parts of the operon. Explain your rationale. And we have a couple of different symbols here. AE means active enzyme. IE means inactive enzyme, I guess. Let me look and check. Yes, inactive enzyme. And what's the next one? NE. No enzyme. Okay, now that's the setup to the problem, and I should probably copy and paste the data into the screen here so we can work through the problem. Okay, so I'm going to copy this in, paste it in, I mean. Okay, it's a little too high here. Kind of want to be down here. Okay, here we go. Okay, so we're working, so we have a number of different strains here. 1, 2, 3, 4, 5, 6, 7, 8, 9, and let's just start by looking at the first strain. So the first strain is wild type for all the different genes, A plus, B plus, C plus, D plus. Remember, we don't know which is the repressor or the promoter or a structural gene operator. We don't know that yet. But when this molecule, this theoretical molecule, is present, and let me take a look at the question again. This operon is concerned with the metabolism of a theoretical molecule. Okay, so we're going to guess that means breaking it down. This molecule is breaking it down. So metabolism is kind of a generic term. It could mean making it or breaking it down. Let's assume that they mean breaking it down. And then if they don't mean that, we'll figure it out eventually, and we'll go back and fix it. But let's assume that they mean catabolizing, breaking down the molecule. Okay, so when this theoretical molecule is present, the enzymes are active. Or we're going to guess this means the structural enzyme, the structural gene. This would be like the equivalent to beta-galactosidase. in the lac operon. This is it's active in the normal case when the TM is present. It's not active, or there's no enzyme, when it is not present. So this is a lot like the lactose system. So when we add lactose to bacteria, the lac operon is turned on. So let's assume that the theoretical molecule is similar to lactose. Okay, so the molecule is present. The structural gene is transcribed and is active. Okay, so we are going to assume that the operon is inducible, like the lac operon. If it was repressible, like the trip operon, right? When trip is present, well then the structural genes are not made. So that's trip is like a repressible operon. This is more like the lac operon. You add the effector molecule, in this case TM. In the case of the lac operon, it's lactose. You add those, and the structural genes are produced, transcribed. Okay. So the first part of the question is done. First, decide whether the operon is inducible or repressible. Okay, inducible. Great. Now let's go on to the next part. We're going to assign A, B, C, and D to the 4 parts of the operon. So let's take a look at the first one here. A, so we have another strain of bacteria, and this strain A is mutated. And when A is mutated, the structural gene is present and active whether or not the molecule is there. Okay, so this sounds like we can have either a mutation in the operator, right? Okay, so I'm going to diagram the theoretical operon, where we have the promoter, we have the operator, we have the structural gene, which I'm going to name S. I'll name it struck. And we have the repressor over here. Now, the operator should have a binding site for the repressor. And so a mutation here or mutation here would both lead to the condition where the structural gene is transcribed, whether or not the molecule is present. So let's assume the repressor has this shape when the theoretical molecule is not around or is not bound. This is our TM down here, theoretical molecule. Okay, this normally binds here and prevents transcription, but if the theoretical molecule is bound, it can't bind. But if this was mutated in a way where this can't bind here, well then this would always be transcribed, whether or not this theoretical molecule is present, right? Okay, so we can have either mutation here or here. So A equals or is equivalent to either the repressor or the operator, because mutations in either of these can lead to the situation where the structural gene is made in whether or not the effector molecule TM is present. Okay, so we got a little closer. That means the promoter has to be either B or D, and the structural gene has to be B or D. Okay, let's take a look at this one. B minus now. Okay, this is a strain where everything is normal except for gene B. And when gene B is mutated, we have no active enzyme being made in either condition. Whether or not the theoretical molecule is present or absent, there's no enzyme being made. So B could be a mutation in the promoter, right? Because RNA polymerase could not bind. Or it could be the enzyme. Yeah, so B could be the promoter or the enzyme too. Well, I think then we would say inactive enzyme. But okay, let's assume we can't tell yet with B. B could either be the promoter. I think it's probably going to be enzyme because they specifically differentiate between inactive enzyme and no enzyme. And they're saying no enzyme. So in this case, I think it's going to be the promoter, but let's hold on a second. Okay, so let's take a look at the next one. Okay, here, C is mutated. Everything is normal, but C is a mutant. And when the molecule is present, we have inactive enzyme. And when the molecule is absent, we have no enzyme. Okay, so it looks like everything is functional. Everything is working as normal. We add the theoretical molecule. The structural gene is transcribed, but it is inactive. Okay, so how can we end up with an inactive enzyme? Only if this is mutated. So this gene is mutated. So C has to equal the enzyme. Okay, so I think everything is starting to come together here. Okay, so you may have noticed that the screen jumped a little bit. I tried to go through and work with these little, with the complicated bacterial strains, genotypes that we're seeing at the bottom of this table, and it got a little confusing, so I'm going to start over and try to do a better job this time. So we've already seen that C equals the, or shown that C is the enzyme, most likely the enzyme, which means B has to be the promoter. And now we need to figure out if A is either the repressor or the operator, and then same thing for D. If A is the repressor, then that means D is the operator. And it could be the other way around. So let me just diagram what we know so far. So we have a repressor there. We have, and these might not be right next to each other, promoter, operator, and the structural gene. Okay, so struct, operator, promoter, and repressor. We know the repressor is, we know the promoter is B, and we know the structural gene is C. Okay, so let's take a look at our first complicated genotype here. Now this bacterium has a plasmid and a normal chromosome. So we'll say this is the chromosome over here, and this is the plasmid over here, and it looks like they have complete operon on both the chromosome and the plasmid. So I'll diagram this over here. So P, O, S, and R over here. This is B, this is C. And on the plasmid, they have wild-type copies of A, B, C, and D. Again, we don't know what A and D go to yet. And on the chromosome, they have a mutant A. And with this bacterial strain, whether or not, when the theoretical molecule is present, we are expressing the enzyme. So remember, the normal condition is that we should not express the enzyme, should not make the enzyme when the theoretical molecule is absent. But in this strain, we make it. So to me, this is telling me that we have some sort of mutation, perhaps on the operator here. So A is mutated over here on the chromosome. It's not mutated over here. And if A is the operator, and the operator binding site for the repressor binding site on the operator is mutated, well then RNA polymerase is going to come in here and it's going to bind to the promoter and transcribe through the operator, transcribe that structural gene, and the enzyme will always be active whether the theoretical molecule is present or not. So this one, even though A is positive over here, it doesn't matter. It means this one can be blocked by the repressor when the molecule is absent, right? But it doesn't matter if this one can be blocked because this one cannot be blocked. And they're both in the cell. So this one's always going to be on if A is the operator and the A in the operator is mutated. Okay, and this one would have no effect. Even though this one's working normally, the enzyme's still being expressed from this copy of the operon over here. So it's looking like A is the operator. And we can look at the others to make sure that that is the case. So A is the operator and D is the repressor. So we have our model now, and let me cut this stuff out of here and make sure the other three strains are consistent with our model of D as the repressor, A the operator, B the promoter, and C the enzyme. Start by diagramming. So this is the chromosome copy, this is the plasmid copy, repressor, promoter, operator, and structural enzyme. Repressor, promoter, operator, and structural enzyme. Repressor is not usually part of the operon. And we have D, A, B, A, C. D, B, A, C. Now in the second complex bacterial strain, everything is normal on the normal chromosome, on the E. coli chromosome, except B. And everything is normal over here. So, and in this case, everything works normal, okay? Theoretical molecule is present. We have active enzyme, or always active enzyme, or just AE means active enzyme. And when it's absent, when TM is absent, we have no enzyme. Okay, good. Now let's see if everything makes sense here. So when the theoretical molecule is present, let's see, the repressor from both of these is completely normal. So D is the repressor, it's completely normal. The molecule is present. We get the allosteric shift. This can no longer bind the operator, which is normal in both cases. So RNA polymerase can come in and transcribe where? Well, it can transcribe from the copy on the plasmid because the promoter is not mutated. We can transcribe and we can make the functional structural gene, which is transcribe the gene which produces the enzyme. So that makes sense that this strain produces the enzyme when the molecule is present. It doesn't matter that this one doesn't work. So RNA polymerase cannot bind to this promoter because the promoter is mutated. It doesn't matter because this copy of the promoter is good, this copy of the operator is good, this copy of the structural gene is good. So the operon on the plasmid is making the enzyme for us when the molecule, theoretical molecule, is present. So what about when the theoretical molecule is not present? Let's see how far back we can go. Okay, great. Okay, it's not present. Perfect. So this can bind here. The repressor can bind. This can bind here. Both repressors can bind to either one, doesn't matter. But both operators will be bound by the repressor. RNA polymerase cannot bind to this promoter anyway, so okay, it's not going to bind there, and this isn't going to be transcribed. RNA polymerase can bind to this promoter here because the promoter is wild type, but it's going to be blocked, and no enzyme is going to be made, and that's what we saw up here. Okay, so far our model is completely consistent with all of the data. So we just need to check one or two more. Okay, and I think the easiest thing to do here is to get rid of all that, get rid of that. Nope, that's not that easy because I got rid of some of the table, and that's not good. So let's get rid of that. Okay, we can still see all the data. Okay, quickly diagram again. Let's hope the model's right, because I hate to go back and have to do this again because we didn't have all the data and the model's wrong. But let's see here. Okay, chromosome copy of the operon, plasmid copy of the operon, repressor is not part of the operon. Okay, R, P, O, S, R, P, O, S for repressor, promoter, operator, structural gene, in case you're just skipping ahead and you didn't pay attention to the first part of this video or walkthrough. Okay, then this is gene D, this is gene A, I made the mistake again, this is B, this is A, this is C. So this is our working model, and so far it is completely consistent with all of the data. Let's check this genotype, this strain right here, and its phenotype. So this strain, the copy of the operon on the chromosome, is perfect except for C. So we have a plus here, plus here, plus here, and the enzyme is mutated, and the copy on the plasmid is perfect. Everything's perfect. Okay, so when the theoretical molecule is present, whoa, we have something weird going on here. Interesting. Okay, theoretical molecule is present. We get active enzyme and inactive enzyme. Okay, theoretical molecule is absent. We get no enzyme. Okay, does this make sense? Well, okay, A is normal, the operator is normal in both cases, so we're going to put the repressor binding site here. Let's do the theoretical molecule absent first. So when the theoretical molecule is absent, the repressor is going to be bound in both cases here. As a result, RNA polymerase is going to try and bind. Oh, it's going to bind to the promoter, but it's not going to be able to transcribe the structural genes in either case because the polymerase is blocked by at the operator. Okay, so now what we're going to do is we're going to consider this case when the theoretical molecule is present. It's binding to the repressors in the cell, and the repressors are undergoing that allosteric shift, and now they can no longer bind to the operators. I can erase those here. Get my pen. Okay, allosteric shift cannot bind the operator now. And looks like it's bound, but it's not bound there. Okay, they're not bound. Now polymerase can transcribe. In this case, it's transcribing a mutated structural gene, so it's making, and that's going to be translated into the inactive enzyme. And this one over here is being transcribed into the normal functioning structural gene, and that's going to make our active enzyme when the theoretical molecule is present. Okay, great. So far, our model is completely consistent with all of the data. Now we have one more to go. Let's hope it is also consistent with that final strain. Okay, as before, I think it's best that I just remove that and quickly diagram operon in both on the E. coli chromosome and on the plasmid. Okay, you should have this memorized by now too. So 4 genes on the operon, 4 on the plasmid, repressor not necessarily part of the operon, promoter, operator, structural, repressor, promoter, operator, structural. Our model says this is D, B, A, C, D, B, A, and C. Okay. Now let's see, in this case, D is mutated in this string, so D is mutated over here, and everything else is normal. Okay, well in this problem, the plasmid had a complete, in each case, in each bacterium, each strain with the plasmid was always completely normal. So each component was wild type when on the plasmid. So in this last strain, it appears that what we think is the repressor is mutated. Okay, so I'm going to put the operator binding site, the repressor binding site on the operator is intact in both. This repressor over here should be normal. Well, let's not look at that first. Let's look at what happens when the theoretical molecule is absent. So theoretical molecule is absent. We get no enzyme being made. And when the theoretical molecule is present, we have active enzyme. Okay, now let's go under, let's assume that the theoretical molecule is absent. Okay, so this repressor here, it makes a normal repressor. So the theoretical molecule is absent, so it's not bound here, and the repressor can bind the operator. And as a result, RNA polymerase cannot, well, it can bind the promoter. If it tries to transcribe, well, it's not going to be able to. It's blocked by the repressor. However, this one over here, okay, this is mutated. So when we have the little D minus, a repressor minus, we usually assume it looks like this. So even when the effector molecule, TM, is not bound here, it cannot bind the operator because it's mutated. So you would think that RNA polymerase would be able to bind to the promoter and transcribe this. But, however, these repressor molecules, they act in trans, right? What does that mean? It means it can be transcribed from one location and it can act on another DNA molecule. So here, and it can act on its own DNA molecule and it can act on others. So we have the repressor suppressing this one, but it's making more than one suppressor. It's making lots of suppressor molecules. And this repressor is normal, and it can also bind here. So it can do the repressing. So even though this repressor is mutated, as long as the cell has one copy of the repressor somewhere, it can bind to all of the operators and suppress expression of the operon, all the operons. Okay, so that makes sense that no enzyme would be expressed in this case. When the theoretical molecule is present, well, okay, what's going to happen? How far can I go back? Okay, let's see. Theoretical molecule is present. This is going to bind here to this good repressor. It's going to cause the allosteric shift. It can no longer bind. Can no longer bind the operator. Get the pen. Okay, so RNA polymerase can come through and transcribe and make the structural gene, leading to active enzyme when the theoretical molecule is present. And also I should add that the theoretical molecule can also bind to the mutated one, but it doesn't matter because this one can never bind in the first place. So this is going to be transcribed because this one, which can bind to the operator sometimes, undergoes the allosteric shift when the theoretical molecule is present and can't bind to this operator and can't bind to that one. Okay, so all of our model, where D is the repressor, A is the operator, B is the promoter, and C is the enzyme, is perfectly consistent with all of this data in the table. Okay, so we're done with this problem. And that's it for problems from this week. So if you have any questions, make sure you come see me. Okay, thanks. See you in class on Tuesday.